Today I am going to explain about topic of amino acids. So amino acids are simple organic acids. So amino acids are simple organic acids. Amino acids are simple organic acid. These amino acids contain carbon, hydrogen, oxygen and nitrogen. So amino acids contain carbon, hydrogen, oxygen and nitrogen. Some amino acids also contain sulfur. So sulfur containing amino acids, these are cysteine and methionine. So more than 200 types of amino acids are present in cytoplasm of the cell. So these are inactivated condition, free in a condition. So in out of 200 amino acids, so 20 types of amino acids are participate in protein synthesis. So these amino acids are generally crystallized solids condition. So crystallized solid condition. These are not soluble in organic solvents, soluble in water only. So amino acids are crystallized solid condition, free state in cytoplasm of the cell. These are water soluble condition, then organic acid insoluble in a condition. Then these amino acids are building block for the proteins. So in proteins these are the monomers. So generally proteins are polymer in a condition. So proteins are composed formation by the member of amino acids. So amino acids are building block for the proteins. These amino acids join with another amino acid by the peptide bonds. Nothing but amide bonds. Peptide bonds are amide bonds. H. So first amino acid and second amino acid. So one amino acid esternified linked with the second amino acid by formation of the peptide bond. So this peptide bond is nothing but polymerization. So condensation of amino acid. Peptide bond formation between first amino acid to second amino acid, carboxylic group and amino group. So in carboxylic group OH and H, elimination of a water molecule, that means CO and NH. So peptide bond formation between the CO and NH. So carboxylic and amino group. So peptide bond, amide bond is nothing but polymerization, condensation, so dehydration, elimination of a water molecule. Like this, among the amino acids, number of peptide bonds are formed. So it is nothing but a polypeptide condition. These amino acids stores the nitrogen in the form of amides. So amino acids stores the nitrogen in the form of amides. Amides are nothing but amino acids, but modified, changed amino acids. These amides contains having the more nitrogen compared to normal amino acids. So generally in amino acid, so structure of amino acids. So in middle, so carbon is present. Then one carboxylic group, then then one amino group is present. So generally in normal amino acids, so one carboxylic group, then one amino group is present. So this carboxylic group and amino group attached to same carbon. So this carbon is known as a alpha carbon. This type of amino acids is known as a alpha amino acids. Alpha amino acids. Then this carboxylic group provides the acidic nature. This carboxylic group provides the acidic nature. This amino group provides the basic nature. So low pH and high pH condition. Addition to this carboxylic group and amino group, then H group and R group also present. So four groups are occupies four valency positions. Four groups occupies in four valency positions. Next one, this R group is a variable group. So R group is a variable group. So in different amino acids, this R group replaced by R could be with different groups. So for example, so this R group could be hydrogen. Best example, glycine. This R group could be hydrogen in glycine. This R group could be methyl. CH3 group alanine. Then R group could be CH2OH serine. So R group would be H hydrogen in glycine, CH3 methyl alanine, then CH2OH serine. These amino acids also substituted to methane. Amino acids also substituted to methane. But in some amino acids, so for example, proline, then hydroxyproline. Proline and hydroxyproline, these amino group replaced by, so amino group replaced by the imino groups. Amino group is replaced by amino group, imino group. This amino acid is known as a amino acids. In proline, in hydroxyproline, amino group is replaced by imino group. This amino acids is known as a amino acids. Next one, glycine. So glycine generally known as a simple amino acids. Glycine is known as a simple amino acids. So H2N. So simple amino acids. In glycine, R group could be hydrogen. That means two hydrogens are attached to same carbon. That's why this is known as a symmetric. So glycine is a symmetric except glycine. All alpha amino acids are asymmetric condition. So except glycine, all amino acids, alpha amino acids are asymmetric condition. So why? Because only one hydrogen group is present in glycine. Only two hydrogens are occupied on a same carbon. So it is a simple amino acids. So amino acids generally in protein laboratory condition. So amino acids generally in proteins laboratory condition. That means coiling in left anti-clockwise condition. Then non-protein amino acids are dextrorotatory condition. dextrorotatory condition. Non-protein amino acids are dextrorotatory condition. Then protein amino acids are levorotatory. Anti-clockwise arranged anti-clockwise condition. Then clockwise condition. Amino acids are present in free state in cytoplasm of the cell. So out of these 200 amino acids, so 20 types of amino acids are participate in protein synthesis. So out of 200, 20 types of amino acids are participate in protein synthesis. These amino acids is known as a protein amino acids. So these amino acids incorporate in proteins based on the triplet codon. That means genetic codon. Genetic codon of DNA or mRNA. So these 20 types of amino acids are incorporation linked arranged in proteins based on the triplet codon of DNA or mRNA. So 20 types of amino acids. So these are phenyl, alanine, then leucine, then isoleucine, then methionine, then valine, then serine, then proline, threonine, then alanine, tyrosine, then histidine, then glutamine, aspartame, lysine, aspartic acid, then glutamic acid. Next one, cysteine, tryptophan, arginine, then glycine. So these are the 20 amino acids are participate in protein synthesis. This is known as a protein amino acids. So these are the coded amino acids. That means based on the genetic code, each amino acid code by the 3 nitrogen bases. This is known as a triplet codon. So these genetic code triplet codon will be useful to understand the arrangement, the sequence of amino acids in a protein, then nitrogen base sequence in DNA and mRNA. So these are the coded amino acids. But in proteins, some non-coded amino acids also present. This is known as a rare amino acids, non-coded amino acids. So this is known as a rare amino acid. These rare amino acids derivatives of the coded amino acids. Rare amino acids are derivatives of coded amino acids. Then these 20 amino acids denoted represents by the abbreviation of 3 letters. Abbreviation of 3 letters. So in name of the amino acids, first 3 letters. So for example, phenylalanine, so PHD, then leucine, NUU, then isoleucine, then methionine, then valine. So first 3 letters. So next one, serine, next one, proline, PRO, then threonine, so alanine. Next one is histidine, then glutamine, aspartame, lysine, aspartic acid, then glutamic acid, cysteine, TR tryptophan, arginine, then glycine. So first three letters in name of the amino acid, but some changes made in some amino acids. So for example, glutamine. So GLN. Why? Because it is a amide. Why? Because it is a amide, modified amino acid. Next one, aspargine. ASN. It is also amide, except amides. In rest of the amino acids abbreviated by three letters. So in a name, first three letters, except these amides. Next, these 20 types of amino acids denoted represents by the alphabets. So different alphabets by the rules and regulation. So phenylalanine abbreviated by the L. Phenylalanine abbreviated by the L. Then sorry. So phenylalanine abbreviated by the F, then leucine abbreviated by L, then isoleucine I, then methionine M, then valine V. Next one, serine S, then proline P, then threonine T, then alanine A, then tyrosine Y, then histidine H, then glutamine Q, then asparagine N, then lysine, K, then D and E. So asparagine denoted represents by alphabet of D, then glutamic acid E. Next one, cysteine C, then tryptophan W, then arginine R, then glycine G. So this amino acid represents denotes by the alphabet symbols. So phenylalanine F, then leucine L, isoleucine I, methionine M, then valine V, then serine S, proline P, threonine T, then alanine A, then tyrosine Y, histidine H, then glutamine Q, amide, asparagine N, then lysine K, then aspartic acid D, then glutamic acid E. Next one, cysteine C, then tyrosine W, arginine R, then glycine G. So alphabets. Next one, non-protein amino acids. Non-protein amino acids. Next one, non-protein amino acids. So what are not present in proteins or not participate in proteins? This is known as a non-protein amino acids. These are more in number. So these are present in a free state or combined condition. These non-protein amino acids are present in free state or combined in a condition. So some non-protein amino acids are found in antibiotics. Antibiotics. So these known as a amino acid analogs. Amino acid analogs. Amino acid. Some non-protein amino acids are found in antibiotics. These amino acid is known as a amino acid analogs. Then some amino acids are participate in biosynthetic process. So for example, citrulline, then ornithine, citrulline and ornithine. These are participate in urea cycle. Citrulline and ornithine participate in urea cycle. Some non-protein amino acids, homo, arginine, then amino butyric acid. So homoarginine, then aminobutyric acid. These are the source of nitrogen. These are the source of nitrogen. Then so gamma amino butyric acid. Gamma amino butyric acid. GABA. So gamma amino butyric acid. These are inhibitory inhibitory neurotransmitter in brain and other parts. So non-protein amino acid gamma aminobutyric acid GABA as a inhibitory neurotransmitter in brain and other parts. So next one, diaminopimelic acid. Diamino so pimelic acid. So this diaminopimelic acid is a intermediate in synthesis of so amino acids are crystallized solid condition. These are water soluble condition, then organic solvent insoluble condition. But these amino acids behave differently in different pH values. Amino acids behave differently in different pH values. So amino acids act as acidic and basic nature if one carboxylic group and one amino group is present. So by changing the pH in solutions, change the amino acid structure also. Then some at neutral pH, at neutral pH values, amino acids are Zwitter ionic condition. So at a neutral pH at 7, these amino acids act as a Zwitter ionic condition. So at a neutral pH, these amino acids are Zwitter ionic forms. So here three types of amino acids. So A, B and C. P is known as a Zwitter ion. That means this has the positive charge and a negative charge. So at one hand positive charge condition, acidic condition, at another and the negatively charged condition and ion condition. If decrease the pH values by increasing the hydrogen concentration, it is nothing but donates the hydrogens. So it is a cation condition at a low pH, positively charged condition. Where at the high pH values, so increase the pH values, lost the hydrogen ions. So it is a negatively charged condition. This form is known as a Zwitter ionic form. So isoelectric point is also known as a isoelectric point. That means the properties of the amino acids are ionizable of NH2 and COO groups. In solutions at different pH, change the amino acid structure. Is known as a Zwitter ionic forms. At neutral pH, amino acids are Zwitter ionic condition. That means act as a acidic and basic condition, as the positive charge and negatively charged condition. So here B is known as a Zwitter amino acids. So at a low pH is a cation, positively charged condition. At a high pH anion, negatively charged condition. So properties of amino acids are ionizable nature of amino group and carboxylic groups. Next, I am going two types of amino acids are present. In this 20 types of amino acids participate in protein synthesis. So generally plants have the capability to synthesize all these 20 types of amino acids from the raw materials. But animals not have the capability to synthesize all these 20 types of amino acids from the raw materials. So which amino acids animals cannot synthesize from the raw materials? These amino acids is known as essential amino acids. So these amino acids animals cannot synthesize from the raw material. It is known as essential amino acids. So these are phenylalanine, then leucine, then isoleucine, methionine, then valine, lysine, then tryptophan. So these 7 is known as essential amino acids in animals. But in human beings, adult human beings, Theo9 is known as a essential amino acids. Children's histidine, then histidine and orchinine. So these also requires at a particular stage for the particular function only. This is known as a semi essential amino acids. This 3 is known as a semi-essential amino acid. So based on the function, these amino acids are classified into essential amino acid, semi-essential amino acid, then non-essential amino acid. These essential amino acids animals cannot synthesize from the raw material. For source of these amino acids is a diet. So diet is a source for these amino acids. Basically 7 phenylalanine, leucine, isoleucine, methionine, valine, cysteine and tryptophan. Then adult human beings. In adult human beings, amino acid Theo9. Next one in children histidine and orchinine. These are required in particular stage only. That's why this is known as a semi-essential amino acid. So in animals, totally 10 amino acids are essential condition. In remaining 10 is known as a non-essential condition. Only in animals, but plants have the capability, have the ability to synthesize all these 20 types of amino acids from the raw materials. Animals not have the capability to synthesize these 10 amino acids. So these mandatory of 7, then 3 is a semi-essential condition. So 10 is known as a essential amino acid, remaining 10 is known as a non-essential amino acids. Then next one is a lipids. So lipids are small or simple fatty acids. So lipids are simple fatty acids of alcohol and other substances. So lipids are simple fatty acids of alcohol and other related substances. These lipids are water insoluble condition. These are soluble in organic non-polar solvents. So these are acetone, then benzene, ether, then chloroform, etc. So acetone, benzene, ether, chloroform, then other organic solvents. So lipids are water insoluble condition. These are soluble in non-polar organic solvents like chloroform, ether, benzene, acetone, etc. Next one, these lipids also contain carbon, hydrogen and oxygen. So lipids contains the carbon, hydrogen and oxygen. Compared to carbon, hydrogen, oxygen is a very less in a condition. Next one, these lipids also contains the phosphorus, then sulfur and nitrogen. These lipids also contains the phosphorus, sulfur and nitrogen. Then lipids. So lipids are classified three types. These are simple lipids, then compound lipids, then derived lipids. So lipids are classified into three types: simple lipids, then compound lipids and derived lipids. So simple lipids only contain this alcohol and fatty acids. So simple lipids contain fatty acids and alcohol. So examples are fats, then sugaring, then cutin and wax is an example for simple lipids. Then compound lipids, addition to these fatty acids and alcohol, some side chains are present like a phosphorus, phosphoric acid and other substances. So compound lipids has side groups along with these fatty acids and alcohol. Examples are phospholipids, then sphingolipids, then glycolipids, then lipoproteins, etc. So phospholipids, then sphingolipids, glycolipids and lipoproteins. So lipoproteins means lipids and protein, then carbohydrates and lipids, phosphorus and lipids, etc. Next one, derived lipids. Derived lipids is nothing but lipids are lipid like structures, maybe derivatives of lipids. So examples: sterols, then terpenes, then prostaglandins. So sterols, terpenes and prostaglandins. So sterols are cholesterol. So cholesterol is the best example for sterol. Then first one coming to simple lipids. So these simple lipids contains the fatty acids and alcohol. So fatty acids are simple lipids. These fatty acids contains the carboxylic group. These carboxylic group attached to R group. So carboxylic group attached to R group. So generally R group is a variable condition. So this R group in fatty acids would be methyl group, then CH2, C2H5 or CH2 condition. So methyl condition, then methyl condition, then methanol condition. So fatty acids contains the carboxylic group. This carboxylic group attached to R group. So it is a variable group. This R group could be CH3, C2H5, CH2. So methanol, ethyl and so methanol condition. So methyl group, ethyl group, then methanol groups are present. Then these fatty acids are saturated, then unsaturated. These fatty acids are saturated fatty acids, then unsaturated fatty acids. So in saturated fatty acids, double bonds are absent in carbon chains. So double bonds are absent in carbon chains. So between the carbon double bonds are absent, maybe single bond are present. So in saturated fatty acids, double bonds are absent in carbon chains, maybe single bond is present. That means the carbons are completely saturated condition. Then saturated fatty acids mainly present in animals. Saturated fatty acids are mainly present in animals. Then at a at normal temperature, the saturated fatty acids are solid condition. Solid condition. At a normal temperature, these are saturated fatty acids are solid in a condition. Then the saturated fatty acids increase the cholesterol levels. So the saturated fatty acids increase cholesterol levels. The saturated fatty acids increase the cholesterol levels. So saturated fatty acids mainly present in animals. They are double bonds are absent in carbon chains. Carbons are completely saturated condition. At a normal temperature, these may exist as a solid in a condition. These may increase the cholesterol levels. Then unsaturated fatty acids. So in this double bonds are present in carbon chains. So more number of double bonds are present. Carbon double bonds are present. These are mainly present in plants. So unsaturated fatty acids mainly present in plants. So at normal temperature, these are exit as a liquid form. These are exit as a are present as a liquid form. Generally, unsaturated fatty acids low the or decrease the cholesterol levels. Lows or decrease the cholesterol levels. Then examples of saturated fatty acids. So stearic acid, then palmitic acid. So examples for the saturated fatty acids are stearic acid and palmitic acids. So generally in fatty acids hydroxyl group also present. So length of these fatty acids generally 4 to 24 carbons are present. In fatty acids along with hydroxyl group carboxylic group 4 to 24 carbons are present. In normal fatty acids so generally 16 to 18 carbons are present. 16 to 18 carbons are present. So in stearic acid 18 carbons, then in palmitic acid 16 carbons are present. So CH3, then COOH. So 14 COOH. So along with the carboxylic group in stearic acid 18 carbons are present. Then in palmitic acid along with the carboxylic group 16 carbons are present. Next one, unsaturated fatty acids. Examples of unsaturated fatty acids. So first one, so linoleic acid, then linolenic acid, then oleic acid, then arachidonic acid. So examples of unsaturated fatty acid are linoleic acid, linolenic acid, then oleic acid, then arachidonic acid. Then in oleic acid, so single carbon double bond is present. Then lenic acid, two carbon double bonds are present. In this three carbon double bonds are present. In the four carbon double bonds are present. So it is a 18 carbon, it is 18 carbon, 18 carbon, then 20 carbons are present, including carboxylic group. So examples of unsaturated fatty acids are oleic acid. So single carbon carbon double bond is present, so it is a 18 carbon condition including carboxylic group. Then linoleic acid, 2 carbon carbon double bonds, 18 carbons including carboxylic groups. Then linoleic acid, 3 carbon carbon double bonds, including carboxylic group. Then arachidonic acid, 4 carbon carbon double bonds are present, 20 carbons are present, including carboxylic groups. So generally, so plants have the capability to synthesize these unsaturated fatty acids. Plants have the capability to synthesize this unsaturated fatty acid, but animals not have the capability to synthesize this unsaturated fatty acids. Why? Because in animals fatty acids are saturated condition. Next one, another simple lipid is a glycerol. Another simple lipid is a glycerol. So glycerol is a trihydroxypropane. So another simple lipid is a glycerol. So glycerol is a trihydroxypropane. CH2OH, CH2OH, then CH2OH. So glycerol is a trihydroxypropane condition. But some lipids may contain both the fatty acids and glycerol. Some lipids may contain both the fatty acids and glycerol. In that lipids, these fatty acids are esternified with the glycerol. That means linked with the glycerol by the ester bonds. So CH2O and CR1. So one first fatty acid. So first fatty acid esternified with the glycerol. This known as a monoglycerides. Monoglycerides. So in monoglycerides, one fatty acid esternified with the one glycerol. Next one, CHOCR2. That means second fatty acid esternified with the glycerol. This known as a diglycerides. Diglycerides, monoglycerides and diglycerides. Next one. So third one, triglycerides. Triglycerides, monoglycerides, diglycerides and triglycerides. Some lipids may contain glycerol and fatty acids, simple lipids. In these simple lipids, these fatty acids are esternified with the glycerol. That means linked with the glycerol by the ester bonds. So one fatty acid with the glycerol, monoglycerides, two fatty acids with the glycerol, diglycerides, then three fatty acids with the glycerol, triglyceride condition. Then based on the melting points, these lipids may be fats and oil condition. Fats and oil condition. So generally these oils as the lower melting points. Oils as a lower melting point. So due to this one, oils remains as a oil condition. So generally oils remains as a oil like a liquid condition in winter season. So oils as a lower melting points, because of this remains as a oil like a liquid phase in winter condition. Next one is a compound lipids. Example, phospholipids. Phospholipids, best example is a phospholipids. That means some lipids may contain the phosphorus and phosphoric acid related substances. So which lipids contain phosphorus or phosphoric acid? These lipids is known as a phospholipids. These phospholipids mainly present in cell membrane. phospholipids are mainly present in a cell membrane. These phospholipids is a nothing but triglyceride. It is a nothing but a triglycerides. Phospholipid is a nothing but a triglycerides. So this phospholipid is a nothing but a triglyceride. That means so first fatty acid, then second fatty acid. So here third fatty acid. But the third fatty acid is replaced by phosphoric acid. So phospholipids are triglycerides. But the third fatty acid is replaced by phosphoric acid. These phosphoric acid link with the nitrogen. Link with the nitrogen group. This known as a choline. Nothing but a lecithin. Nothing but a lecithin. So these phosphoric acid link with the nitrogen group side chain nitrogen group like a choline. So also known as a lecithin. So additional nitrogen group. So phospholipids, lecithin. So lecithin is a phospholipids mainly present in a cell membrane. These phospholipids are amphipathic condition. Amphipathic nature. So these phospholipids are amphipathic nature. That means carry the hydrophilic polar then hydrophobic non-polar ends. So phospholipids are amphipathic condition. That means carry the both hydrophilic polar ends then hydrophobic non-polar ends. Phospholipids best example is lecithin. Lecithin present in a cell membrane. Then another lipids are derived lipids. So derived lipids are lipid like structures. So best example sterol or terpene, then prostaglandins. So cholesterol. So cholesterol is an example of a derived lipids. So cholesterol generally present in plants and animals. So in plants, Solanum tuberosum. So in plants, Solanum tuberosum. So generally in animals, so these are present in free condition or branched condition. So cholesterol, so it is a structure of a cholesterol.